Good morning, everyone, and welcome to our first homegrown research webinar for 2024. Today, we will be talking about fertilizer management. My name is Eileen Beaton, and I am the Crops Extension Specialist with Saskatchewan Agriculture in Weyburn, and I will be chairing today's webinar. CEUs are being offered for certified crop advisors on this webinar. To get credit, you need to attend the entire live event, and there will be a QR code at the end of the presentations to collect credit. If you do end up missing the code but did attend live, please email us afterwards and we can work with you to get the credit. Before we begin with the formal presentations, we will begin with an introduction to the Homegrown Research Series. So you may be asking, why are we calling this the Homegrown Research Series? This is because all the research presented today and on upcoming webinars is from the research sites involved in the Agri-ARM network across Saskatchewan. It is all local research that can be directly applied on your farm, and you'll notice all the logos on the screen here for all the sites. What is Agri-ARM? Agri-ARM is a network of producer-directed, applied research and demonstration organizations located across Saskatchewan. All operating individually, each of the sites strive to collaborate on a broad range of projects, generating results relevant to growers over a wide area. Agri-ARM sites receive funding through the Saskatchewan Ministry of Agriculture, as well as project-based funding from governments, commodity groups, and private industry. What do Agri-ARM sites do, and why does it benefit Saskatchewan producers? Agri-ARM is a one-stop for research, demonstration, and extension activities across Saskatchewan. Specifically, the sites work to increase adoption of new agricultural technologies and practices, provide farmers with more tools for decision-making, test, evaluate, and demonstrate potential new technologies and practices, and provide unbiased, third-party results at field days, producer meetings, workshops, as well as online. The sites provide reliable results across a wide range of soil and climatic conditions, local data relevant to local farms, comprehensive support for adoption of new technologies, and support for development and validation of new technologies. We encourage you to visit the Agri-ARM website at the URL shown here on the screen. Here you can find more information about the organization, upcoming events, current projects, fact sheets, or research updates. The website also contains contacts and more information for each site. Agri-ARM focuses on local producer needs and is always looking for new ideas. You can submit your ideas by reaching out to the site nearest to you or by using the submission form on their website as shown here. We encourage you to visit the website for your local Agri-ARM site, where you will notice research updates, contact information, and ways to follow them on social media. Sites often host field days or winter meetings to share research results. Here you will notice dates for each site's upcoming summer field day. Contact your local Agri-ARM for more details. In the next few slides, I'm going to highlight each of the sites we have representatives from today. Jessica Enns is joining us from the Western Applied Research Corporation located in Scott. WARC is a nonprofit organization that is governed by a board of directors consisting of Northwest Saskatchewan producers. They work in partnership with AAFC at the Scott Research Farm. Also speaking today will be Brianne McGuinness, Operations Manager with the Northeast Agriculture Research Foundation. NARC was established in 1997 to coordinate and support crop production research activities in northeastern Saskatchewan. It is a not-for-profit organization that consists of a farmer board. It is also affiliated with AAFC Research Farm at Melfort, Saskatchewan. Today we also have Amber Wall, who is joining us from the Wheatland Conservation Area located in Swift Current. WCA was established in 1983 by producers concerned about soil degradation, with a special emphasis on soil salinity. They take a proactive approach to member concerns and have adjusted programs and mandates to include applied research and demonstration of advanced cropping systems. Mike Hall is joining us from the East Central Research Foundation located in Yorkton. ECRF was established in 1996 and is a nonprofit organization whose mandate is to conduct crop production research and extension activities for the agricultural industry in East Central Saskatchewan. In 2013, a memorandum of understanding was signed between the ECRF and Parkland College to share resources and jointly pursue agricultural research and extension. The partnership has enabled Parkland College, which is now Suncrest College, to become the first regional college in Saskatchewan to undertake an applied research program. Our final speaker today will be Kim Stonehouse, who has been a crops extension specialist at our regional office in Tisdale since 2009. On the slide here, you will see our 10 regional offices located across the province that hold a multitude of specialists to help you with your agriculture needs. I'll also take this time to briefly mention the 2024 Crop Diagnostic School, which will be held in person on July 24th and 25th in Melfort in partnership with NARF. So keep watching our social media pages for details on how to register. Along with this information, we do have additional resources for everyone under our handout sections in the chat box area of the toolbar. Resources will be shared along with the recording after the webinar is complete. Today, we will have a 15-minute presentation from each speaker, followed by a question panel with all the speakers. The main hub for today will be your webinar screen. And as mentioned, we will share relevant downloadable handouts with you in the chat box area. The entire session will be recorded, so if you miss anything or wish to revisit any of the presentations, you'll be able to. Recording links will be sent out in the next few days in a follow-up email. An evaluation form will also pop up at the end of the webinar this morning, so please fill this out because this really helps us in determining what topics that you want to learn more about, and it helps us prioritize research needs for the future. So just a few housekeeping notes to keep in mind during the webinar. If anybody does have any questions during any of the presentations, you can type them into the question box in the bottom right-hand side of the control panel. We will try to get to as many of your questions as possible, and your questions will remain anonymous, and they're only going to be sent to me. So during the presentation, you will be muted so that we can ensure there is no feedback that will interfere with the audio. So with that, we will get started. And we're actually going to start with a presentation from Jessica Enns, who has worked as a research manager for the Western Applied Research Corporation located in Scott since 2015. Her topic is demonstrating the efficacy of foliar-applied nitrogen-fixing bacteria for wheat and canola.
Perfect. Thank you so much, Eileen. So, our presentation today is going to be focusing on our homegrown fertilizer management program. As she mentioned, my name is Jessica Enns, and I've been with WARC since 2015. So the first topic we want to talk about is the efficiency of foliar-applied nitrogen-fixing bacteria in canola. The lead principal applicator was Chris Hosaffel. We located this at all 8 Agri-ARM sites. And of course, I need to make a special thank you to the funders. This went through the ADOPT program, through the Sustainable Canadian Agriculture Partnership, as well as financial assistance occurred through SAS Canola. So one of the things we really wanted to dig into when we were talking about this project was, what are the benefits of foliar-applied nitrogen fixating crops in non-host crops? So the first thing that we really wanted to figure out was, why is this so important? Well, nitrogen, as most of you know, is the most commonly limiting nutrient in our production for non-legume crops in Saskatchewan. And currently it's typically the most expensive input. This has rang especially true in the recent years, as we've seen record high nitrogen levels. The other problem that farmers are really struggling with is the increased demand to reduce our nitrogen associated with greenhouse gas emissions. So this is really putting on a lot of pressure to reduce the amount of nitrogen we're putting down, but as well as increasing the nitrogen use efficiency. So not only do we have to reduce what we're putting down, but figure out a way to make it the most efficiently as possible. And while we're doing that, we also need to make sure that we're hitting our yields so that we're making money. And we need to be able to hit those yields so we can feed the world. So that really rang into a problem for a lot of producers. So one of the ways that we're hoping to tackle this problem is through biological products. So what we're hoping to find is, with these biological products, are they able to improve the efficiency and the sustainability of crop production? And this is through the fixation of natural nitrogen. So what we're hoping is that this will be able to subsidize the soil and nitrogen fertilizer that we'd have to apply. And so overall, this will help us reduce the amount of nitrogen we have to put down and improve our nitrogen use efficiency. So that was really the goal out of this project. So the objective here was really to understand, how do these projects, or the program, work under different field conditions and under different soil zones. We focused on 2 different products. We used Envita as well as Utrisha. Now, we do recognize that there's certainly multiple products within the industry that could have been used, but we decided to go with these 2 as we felt that they're the most known to producers. So the 2 was Envita, which is a nitrogen fixing bacterium, and the other one was Utrisha, and this is actually a nutrient efficiency bacterium. So I'm going to go through it and just kind of explain the minute differences between the 2, just so we have a really clear understanding of how these products truly work. So the very first one is Envita. This is a naturally occurring bacterium called Gluconobacterium diazotrophicus, and I will be calling that GD from now on. This bacteria is naturally occurring, and what this does is it creates a biofilm around the bacteria, and this really allows it to penetrate through the plant, through the stomata that we can see here, through the leaf cells, as well as through the roots. And so what this does is it's really able to penetrate through the entire plant. Now, the other really special component of this bacteria is that it is systemic, so it allows it to move through the xylem. So that really allows it to spread throughout the entire plant. And so what we're seeing is, once it moves through the entire plant, it's able to really colonize throughout the entire leaves, the roots, all the leaf hairs. And so this is really important because it gives it the ability to fix nitrogen within the entire system. Now, once this bacterium is colonized, what it does is it basically creates vesicles within the cells. And that's what we're seeing here. Is, when you have to say a vesicle, it really makes you think of something sort of like the rhizobium creating a nodule on your pulse crop. So it's really similar to that. But what this is doing is occurring inside a cell. And within that cell, it's able to produce nitrogen. And it takes the atmospheric nitrogen, or your N2, and it converts it to ammonium. And it produces that nitrogen throughout the plant when it's required. Now, this differs in the way that it produces its photosynthetic energy from Utrisha. So Utrisha actually uses a Methylbacterium symbioticum. And what this is actually a nutrient efficiency bacterium. So it works in a very similar way. You spray it onto the leaves, it penetrates through the stomata, and it enters into the photosynthetic cells, and it colonizes again within the plant. First within the leaves and then in the rest. Now, once it's colonized, it starts to create this bacterium called methylcalobonium. And what this does is that it actually absorbs and reflects the light into the chloroplast. And so this actually boosts the photosynthetic activity and its energy. And when it's able to do that, it activates the nitrogen complex to generate more ammonium from your elemental nitrogen. So they work in a very similar fashion in the fact that they both use the nitrogenase complex to generate ammonium. However, Nutritia basically boosts the amount of energy, whereas Envita actually produces its own nitrogen. So it works in two slightly different fashions. So now that we have a very general understanding of how this works, what we find is we wanted to figure out the best way to apply these bacteriums in different fertilizer categories. So what we did is we broke it down to a low, medium, and high fertilizer applications. So when we did this, low is basically we're going to be taking the background fertilizer levels in the soil, as well as adding additional fertilizer. And so we wanted to treat this as you would typically treat regular canola. So at the maximum was 143 pounds of total N. And then we had it in untreated with the Envita and with Nutritia. We located this at all 8 sites throughout the Agri-ARM district, and we did this because we really wanted to go back to our objective and figure out how does this work in different soil zones, how does this work under different environmental conditions, and so we can really tell for producers, you know, how this works in different areas, and is there a difference in different areas. The first step we did once we had established this trial is we went back and we soil sampled our soils prior to any of the applications because we wanted to know what is our background fertility levels. So the thing you can see here is Scott had around, or sorry, Indian Head had around 18 pounds of N, Melford had 39, Outlook had 18, and then PA here actually had really high levels of background nitrogen. So they actually already had 64 pounds of available N. So they didn't add any additional fertilizer because they already exceeded our low treatment. And then everyone else had relatively low background. Yorkton had a slight increase of around 42 pounds of available N, so they only put down a very minimal amount of urea. This fertilizer was also applied at seeding through the sideband. Now, the other factor that we really wanted to take into consideration was applying it properly. Because this is a living species, we really wanted to make sure we followed the label to a T. So we applied it at the 4 to 6 leaf stage. So this is during the vegetative stage. We wanted to make sure we had really good coverage. So we had 10 gallons an acre. We stored them in a cool room so that the rhizobium weren't stressed. We used distilled water and we applied in the early mornings or on relatively cool days. And this is to allow the bacterium to really be able to penetrate through the stomata, through the leaf hairs, to really improve its colonization within the plant. So we'll get just right down to the nitty-gritty of the results. What we did here is this is looking at the combined data across 5 locations. Now, we did have a total of 8 sites, but we did not have a response to the nitrogen levels at Scott or Prince Albert. And Swift Current actually had hail in this year, so we did exclude that as well. So we wanted to look at the most responsive sites to our nitrogen application. And then within that data, we looked at whether or not it was responsive to those foliar nutrient applications of Envita and Nutritia. So the first thing we really want to point out here is the different levels of statistical difference between the trials. So what we looked at is, for example, at Indian Head, there was no statistical difference between any of the treatments. As you can see here, they all had A's. So if it's an A, and A, that means that there is no statistical difference between any of the treatments. And when we go through all of the yield and all of the locations, we have Indian Head, Melford, Outlook, Redverse, Yorkton. None of them had any statistical differences because they all share the same letter. The only difference we did find was that at Outlook, you can see here, there's a B and an A. So we actually did find that Nutritia did perform slightly better than Envita, but overall it was a fairly minute difference and it was only a one-off, but we did find a slight increase. Now, when we looked at it in terms of an average over those 5 site years, there was unfortunately no difference between the untreated control, the Envita, and the Nutritia. So then the next thing that we investigated is how did these products hold up at the different nitrogen levels? So we had that low and the medium and the high fertilizer that we applied. And so when we looked at that across the different products, we found that there was no significant difference as well. So you can see that there's all Ds and then all Cs and then all A's in every single of those columns, which indicates that there was no significant difference. Now, when we looked at this in terms of seed quality, we looked at our oil and we looked at our seed protein. And unfortunately, we did not find any differences in terms of seed oil at the different locations. So once again, at the low, the medium, and the high, they all shared a similar letter. So that meant there was no differences in the seed oil. And then once again, when we looked at it in terms of seed protein, there was no differences, unfortunately. Now, to kind of wrap up what we saw here, unfortunately, there was no indications of improved nitrogen efficiency to these products. We did see responses to our nitrogen applications, but that was in terms of our urea applications, but unfortunately not to the foliar applications itself. Now, there is quite a bit of research backing both of these products in terms of their efficacy for their nitrogen use efficiency. So that doesn't mean that we won't necessarily see a response if we continue to investigate. Maybe it's a different environmental conditions, maybe it's different timings, maybe it's a different crop. So we're really trying to kind of dig into this as well and to figure out how do we make this work for producers, even though we didn't see anything here. Based on these results, we would also strongly recommend that farmers do not change their nitrogen levels at this time because we just aren't able to confirm that these products are able to improve your nitrogen use efficiency. So keep doing what you're doing. And if you are going to use these products, make sure you use a test strip so you can actually see the difference on your own farm and to really evaluate that efficacy. So now we'll kind of move on to our next project. It was very, very similar. We did the exact same components where we looked at the foliar-applied nutrients, but we did this time on wheat. Once again, Chris Hosaffel was our lead here, and the funding occurred once again through the ADOPT program under the Sustainable Canadian Agriculture Partnership, and we partnered up with Sask Wheat for additional finances. Once again, we had a very similar treatment layout. We did reduce the amount of total nitrogen at each treatment just a little bit lower than our canola, just to really mimic what we typically do in our farming practices. So low was 45 pounds of total N, medium was 89, and high was 134. We used the same two products. We used Envita versus Nutricia, and then we compared that to our untreated control, so nothing was applied on that one. And we took into consideration the same application factors. We applied it once again at the 4 to 6 leaf stage. We used 10 gallons an acre. We stored it in a cool room. We used distilled water, all of those things to really ensure that we had the most efficacious application that we possibly could. Once again, we located this throughout the entire province, utilizing all of the Agri-ARM sites. Now, in terms of yield, when we looked at our spring wheat, we did combine once again across the 5 locations. But where these locations varied from the canola, we actually didn't see a response to our nitrogen applications at PA and at Yorkton, so we pulled that data out. And we also saw a very weak response at Swift Current, and because of the hail damage there, we also took that data out. So what we really wanted to do was focus on the treatments or the locations that had a positive response, and then once again, we dug into that data a little bit more to see if there was a foliar nutrient application effect. So once again, when we look at the data, unfortunately, you can see here that there's A's across the bottom of all of our treatments, which means that there was no statistical differences between any of the treatments at any of the locations, or when you combined it across the average, unfortunately. Then we wanted to look at it once again in terms of our responses at the different nitrogen levels. So we had low, medium, and high once again, and unfortunately, you can see here that the low had all D's, which means that there wasn't any difference. At the medium, we had all C's and there wasn't any difference, and at high, we had a combination of all A's. So unfortunately, we didn't see any response statistically. When we looked at it in terms of grain protein, the first thing that we really noticed we wanted to make sure we could see was the response of protein to our different nitrogen levels, which isn't a surprise to anyone. I think most of us know that when you add more nitrogen, you typically have a linear effect with your protein level. So as we increased our nitrogen, our protein increased, which is what we expected to see. And that's what you really see in this first graph here. Now, the second graph is where we wanted to compare our untreated to our Nutricia, to our Envita. And that's where we wanted to see if those checks made a difference. And unfortunately, at any of 3 nitrogen levels, we didn't see any significant data responses. So once again, there wasn't any typically response to any of the variables that we measured across any fertilizer level or location. We did see a nitrogen response in terms of yield to our added urea, but that unfortunately was it. And once again, there wasn't any response, but we do hope to see positive responses in the future with different crops and under different environmental conditions, possibly different timings. So that is something that we still want to really investigate, because I think that this is a worthy cause. It's just going to require maybe a little bit more finessing. Once again, based on these results, we really don't recommend anyone changing their nitrogen rates, because we can't truly prove that we're having an increased nutrient efficiency with this product. With that, I wanted to say thank you so much for tuning in today and for your time. If you have any questions, please feel free to contact us. And with that, I'll let Eileen take over. Thank you.
Yes, thanks so much, Jessica. As a reminder, if you have any questions today, please enter them into the chat box on the bottom of your control panel, and we'll hold on to them until the question panel with all of the speakers. So thanks again, Jessica, for all that great information. We really appreciate the time you've taken to be here today. So next up, we have Brianne McGinnis, who has been the operations manager for the Northeast Agricultural Research Foundation, located in Melfort since 2018. Her topic is mid-row versus side banding fertilizers in oats. So we will start whenever you're ready, Brianne.
All right, thank you for that introduction. So my name is Brianne McInnes. I've been with the Northeast Agriculture Research Foundation, based just south of Melfort, Sask, for about 6 years now. So we've got a wide variety of different projects going on at our site. We have 45 different projects this past season, but I'm going to focus on one particular project we've been working on the past 2 seasons at our site. Looking at for our management in oats. So the title of the project was For Our Management, the Right Place and Rate for Fertilizer in Oats. So obviously when we're considering the 4R principles, we're looking at the source of fertilizer, how are we going to place that fertilizer, when are we going to apply it, and then what rates are we going to use. So within this project, we're going to focus on placement and then also rate, with a little bit of a consideration for source as well, looking at side banding versus mid-row banding of nitrogen. And then we've also got a bit of a placement effect with phosphorus as well. And then I just want to acknowledge our funders for this project, which was the Government of Saskatchewan's Agriculture Demonstration of Practices and Technologies, or the ADOPT program, funded by the provincial government. And then we're working in collaboration with Sask Oats, with a little bit of co-funding provided to us by Fertilizer Canada. So here we have our treatment list. So we had 12 different treatments for this project. The first factor was looking at nitrogen placement of mid-row banding or side banding. Then we've got a no nitrogen control, and then a comparison of 3 different nitrogen rates, 75, 100, and 125 kilograms per hectare. And then our third factor is phosphorus placement. So we had a no P control, and then comparing seed row placement versus side band placement. So we have a little bit of an unbalanced design for this project, mostly because Sask Oats was wanting to look at this project from the standpoint of what options do you have for fertilizer placement as a producer if you have mid-row banding capabilities versus side banding. So if you're mid-row banding your nitrogen, you likely only have the option to seed row place your phosphorus, whereas if you're side banding your nitrogen, you have that option of either seed row placing or potentially side banding your phosphorus as well. So the years for the project were the past 2 seasons, so 2022 and 2023. This is just a picture of our plots at Melfort on the right there. The sites in the project were Melfort and Scott, so the northeast and the northwest part of the province. Our design was an unbalanced split plot, so within that treatment list, we had treatments 1 to 6 within the first half of the split plot, and then the side banded treatments for nitrogen were all within the second half of the split plot. This design was really just chosen based on the seeder capabilities at both of our sites. The design just made the most sense for ease of operations at seeding time for us. And then we had 4 replications that were fully randomized within each half of the split plot. And our data collection consisted of plant counts, days to maturity, grain yield, test weights, and then seed weights. We're using urea for all of the nitrogen placement options, monoammonium phosphate for phosphorus. I should mention that our phosphorus rate used was 40 pounds per acre. We use CDC Arborg, targeting recommended plant densities of 350 plants per meter squared in both years. And essentially outside of treatment indications, we are just managing for best management practices for herbicides, insecticide, fungicide, etc. So now just a little bit of background on the site info as far as soil characteristics go. So Scott is in the dark brown soil zone. They've got more of a coarse textured soil, sandy loam with a more moderate organic matter content of around 4%. They had pretty low residual N in both years, and then they had low to relatively high phosphorus depending on the year as well. And then they have pretty good soil conditions as far as a neutral pH with pretty low salt content in this specific trial area anyways. And then at Melfort, we're in the thick black soil zone. We have a finer textured soil with clay loam consistency and a relatively high organic matter of 8 to 10% depending on where we're at in our fields. We had moderate to very high residual N depending on the year. In 2022, we were coming out of a drought year and we had some pretty high N in our field, which really diminished the likelihood of the nitrogen response for us. And then we had a little bit more of a moderate level of N in our soil going into 2023. And yeah, we have a pretty relatively neutral pH as well with low salt. As far as the conditions go, no surprise that we were both hot in both years. The difference would be, I guess, a little bit more in precipitation. In Melfort, we had pretty different conditions depending on the year. We were closer to 50% of our average rainfall this past season, whereas last year we were just over our cumulative precipitation. Scott was more comparable in rainfall between the 2 years, having 70 to 80% of their average cumulative rainfall. And then as far as temperature, conditions were similar at both locations. I think the major difference is that we had a more normal spring in terms of the temperature last year, followed by a pretty hot summer, whereas this year we had a really hot spring followed by kind of more average temperatures into the summer months there. So this is just a picture at the Melfort site, kind of showing the vastly different conditions between the years. In 2022 and 2023. These pictures were taken at a similar time near the end of July, so our maturity was very different between the years. On the right here, I would say you could see a little bit of a different maturity response from the control in the middle there to the highest nitrogen rate on the right, but unfortunately, statistically, it was never significant. So now focusing first on the phosphorus effect. So phosphorus never significantly affected plant density, days to maturity, test weights, or seed weights at any of the sites. So this is just showing the general differences in plant density between the sites and years, as well as the days to maturity. Overall plant density was usually pretty good. Scott 2023 is kind of the lowest it ever got, but still a fairly acceptable plant density there. Days to maturity, pretty comparable between the years at Scott, whereas at Melfort we had a pretty big difference in maturity between the years, with 2023 having a pretty early maturity and almost increased by about 20, or I think it's around 20 days, sorry, from 2023 to 2022. Now, as far as grain yield response to phosphorus goes, the only site year where we did detect statistical significance was 2022 at Scott, where we saw a significant response to phosphorus as compared to the no-P control, but there never was a significant difference between the placements. At Melfort in both years, there never was a significant difference between the no-P control and seed placed and side banded, but there was a tendency for average yields to be a little bit higher for seed placed P as compared to side banded P. Now when we look at the effect combined across years and sites, there was a significant grain yield response to seed placed phosphorus as compared to no P. There was a tendency for yields to be greater with side banded P as compared to the control as well, but it was not significantly different. Now just focusing on the nitrogen placement effect, I only included results that were significant. So if it's not included, that's just because there were no significant treatment differences. So at Scott in 2022, they did have a significant reduction in plant density when side banding as compared to mid-row banding, with a loss of about 30 plants per meter squared. And then when we combined nitrogen rate and placement, there was a significant reduction in plant stands when you were side banding nitrogen and pushing high rates. So about a loss of 50 plants per meter squared when going from 75 to 125 kilograms per hectare of N. And then at Scott in 2022, there was a significant increase in the days to maturity with side banding N as compared to mid-row banding N. And then at Melfort, there was a significant grain yield response where side banded had better grain yield as compared to mid-row banding. This is the combined effect. So when we combined all sites and years, there was a significant response where grain yield was significantly increased with side banding nitrogen as compared to mid-row banding nitrogen, but there was also a loss of about 1.5 grams per half a liter with side banding for test weights. And then overall, when combined across years, there was a loss of about 25 plants per meter squared when side banding as compared to mid-row banding nitrogen. Now, when we just focus on the nitrogen rate effect, there was an increase in days to maturity at Scott when increasing nitrogen rate, increasing by about a half a day for each increase in nitrogen. And then as far as grain yield goes, in the first year, there were no significant responses to nitrogen rate. At Scott, overall yield potential was really low in 2022. And then at Melfort, we had really high residual N, which diminished the potential for a significant yield response. Whereas in 2023, we both had significant yield responses at both of our sites, where yield was maximized at Scott with 100 kilograms per hectare of N, and at Melfort, yield was maximized at 125 kilograms per hectare of N. This is the response of test weight to nitrogen. So in 2022 at Melfort, it seemed like nothing we could do could really reduce our test weights, and we had really good test weights across the board, whereas all of their sites and years, there was a reduction in test weight going from the control to that first nitrogen rate of 75 kilograms per hectare. Now, when we're just looking at the side banded treatments for nitrogen rate by phosphorus effect, when we increase nitrogen rate at Scott, plant density is going down. And then at Melfort, when we have seed placed phos in along with high rates of side banded nitrogen, we're starting to see an increase in the days to maturity. And then at Scott for test weight in 2023, there was a significant difference between the nitrogen rates where you were losing about 4.5 grams per half a liter when increasing from 75 to 125 kilograms per hectare, which isn't surprising as we know our test weights often are reduced when nitrogen is applied. So as general takeaways for nitrogen placement, side band was the best for yield. But mid-row was better for having a good stand and having good test weights. For nitrogen rate, high rates were needed to maximize yield, but residual N and yield potential of the site were definitely affecting the response to high rates. And then of course, with added N, there's the potential for lower test weights as well. And then as far as phosphorus placement, seed place did result in the best yields overall, but of course the rate we used was low and was within the seed safe rate. So if you're pushing rates in the seed row, it's hard to say that that would still be the same response, but when you're using a lower rate of 40 pounds per acre, it did result in the best yields. With that, just want to say thank you to our funders, the Adopt program and Fertilizer Canada, as well as our collaborator of Sask Oats. And then also thank you to our staff at NARF and Work for helping us complete this project. And then I just have listed our website. We're on Twitter and YouTube if you want to stay updated on any research we have going on at our site. So thank you.
Thank you so much, Brianne. As a reminder, if you do have any questions today, please enter them into the chat box on the bottom of your control panel, and we will answer them during the question panel. So thanks again for all that great information, Brianne. We really appreciate you coming on to this webinar today. So the next speaker we have today is Mike Hall, who has been the research coordinator with East Central Research Foundation since 2013. And his topic is how changes in nutrient management affect the profitability of oat. So we will start whenever you're ready, Mike.
Hey, good. All right, so I've been the research coordinator for the Yorkton Research Farm, which is a collaborative effort between Suncrest College and the Central Research Foundation since 2013. And if you're wondering who the Suncrest College is, it's an amalgamation between Cumberland College and Parkland College. They joined together. So if you take an informal poll of producers and ask them how much nitrogen they're applying to oats, you often get somewhere in the range of 60 pounds of nitrogen per acre. And a lot of producers will, when you ask them why don't you put more down, they'll talk about issues with lodging, maturity, and low test weight concerns. And those are all valid concerns to be concerned with. But some more recent work from, Bill May and ourselves here at ECRF often find 90 pounds of nitrogen per acre was a more economic rate to be putting down during good conditions, and if your soil test nitrogen is less than 30 pounds per acre or less. Now, some are concerned that they may be asked to apply less nitrogen in the future due to environmental concerns and are wondering how this might affect their bottom line. Well, if we go back to some past research that we've done in Yorkton, here's 2019 with CS Camden. What I'm showing you here is the blue line is the yield response to increasing nitrogen, and the orange line is the test weight. And so as you increase nitrogen here, you can see that the test weight is declining. Now, if you get a test weight that's below 240 grams per half a liter, you're in the discount range for milling. And if you get a test weight below 230 grams per half a liter, you're in the rejection area for test weight for milling oats. So at the time, I used $6 per bushel for oats and $1.33 per pound of nitrogen. Prices have changed since then, but it doesn't change the interpretation of the results too much to today's values. And so we ended up with 90 pounds of nitrogen at this particular site, and variety being the most economic rate of nitrogen to be putting down. If we compare that to Summit oats, did you notice that when I went between the two, there's Camden, and then I'm going to jump to Summit here. Watch how the test weight line jumps up for Summit. And we often see that with Summit, that it has much better test weights than Camden. But Camden is a very popular oat variety to grow still, mostly because it doesn't lodge as much as Summit will. And test weights haven't proved to be too much of an issue at grain millers for the producer. So it's still a good variety to grow, even though we often see Camden being the bottom of the pile for test weight. But here we're looking at Summit oats, and its most economic rate of nitrogen was 94 pounds of nitrogen per acre. So let's say you're putting 90 pounds of nitrogen down, and someone comes along and says you need to reduce that by 30%. What does that do to your bottom line? Well, in this case, it reduced your economic returns by about $15 per acre. But let's say you're more conservative and you're putting 60 pounds of nitrogen down because you risk averse, fear of lodging, there's variability in your field. And now somebody comes and says, reduce your nitrogen rate by 30%. That reduces your net returns by $24. So it reduces your returns even more so if you're not fertilizing aggressively, which intuitively makes sense. So Sask Oats wanted to fund these 4 locations here to answer the question in 2023, what is the impact of reducing N rates by 15 or 30% on the bottom line? So in their trial, we looked at Camden and Arborg, Camden being a popular variety grow and Arborg being one that's up and coming or growing in popularity. We assumed 125 pounds of nitrogen, which also includes what's in the soil, as well as what you're applying with fertilizer, as being our target rate for both of those varieties. And then we looked at a reduction of 15%, which takes us down to 106. And then we looked at a reduction of 30% that takes us down to 88 pounds of soil plus fertilizer N. The soil-only N treatment varies between locations because the amount of nitrogen in the soil varied between locations, and those are listed below. So in this chart, you can see we've got the yield response in blue, and in orange this time, we have the amount of nitrogen that was left over after harvest from each application of nitrogen. So in determining the economics of cutting back nitrogen, in this case, I'm going to assume 80 cents per pound of nitrogen and $5.25 per bushel of oat. And that's based on the 2024 Saskatchewan Crop Planning Guide. So if we do our first reduction here, we've got, let's hang on here. If we do our first reduction here, 15%, of nitrogen. And this particular curve that you're looking at here is for all the dryland sites. So we've taken Outlook out of this data set here because Outlook was an irrigation site and had a much better nitrogen response curve. For the dryland sites, they were pretty dry last year. Dry conditions were fairly widespread last year. So when we reduced nitrogen rates by 15% here, we're actually gaining money here. We're gaining anywhere between $28 and $17 per acre, depending upon whether you'd like to focus on the data points or whether you want to take your economics based on the response curve. So, you know, we're making money by cutting back nitrogen in this particular case on these dryland sites during the dry year. If we reduce the nitrogen further to a total of 30% reduction, now we're kind of a break-even proposition here. We're either losing a bit of money or gaining a bit of money, depending upon whether you look at the data points or the response curve here. So basically, the most economic rate of nitrogen for these sites last year was 88 pounds of nitrogen per acre, which includes what was in the soil. If we look at the nitrogen that's left over at the end of the year, we can see that as we increase nitrogen rate early on in the year, we increase the amount of nitrogen that was detected with soil tests post-harvest. So 85 pounds of nitrogen have been applied here in total, and it's increased our soil level of nitrogen by 25 pounds. So you can safely say 25 pounds of nitrogen didn't make it into the crop, obviously. But it would have been more than that because we can't account for all the other mechanisms of nitrogen loss in the system. But because of the dry conditions, a lot of the nitrogen that we did apply just didn't go to crop productivity. If we look at Outlook now, you can see it has a steeper response curve to added nitrogen. And if we look at a 15% reduction in nitrogen here, we are gaining money again at this site. So the highest rate of nitrogen was not the most economic rate at even the irrigated site. However, if we do another decrease in nitrogen for a total of 30%, now we're starting to lose money. We're losing anywhere around 20 bucks per acre. And so the most economic rate of nitrogen at the irrigated site was 106 pounds of nitrogen of soil plus applied fertilizer. If you look at the orange line here, you can see that there isn't much nitrogen left over at the end of the year. And that was regardless of the rate of nitrogen that was applied at the beginning of the year. So this is in contrast to the dryland sites. And the reasons for this is a lot more of the nitrogen that we applied went towards increasing crop yield, but it's also under irrigation. So there are more nitrogen losses occurring here too. So nitrogen's being lost to leaching or denitrification from the system. So conclusions, the impact of cutting back on nitrogen by 15 to 30% depends on the following. Are you fertilizing aggressively, like 125 pounds of soil plus fertilizer N? If yes, then cutting back will cost you about 15 bucks an acre under ideal conditions. If you're fertilizing less aggressively, say 90 pounds of nitrogen of soil plus fertilizer N, then yes, cutting back will cost you more, like up to $24 per acre under ideal conditions. But if you're experiencing a drought, then cutting back on nitrogen is actually saving your money in our example. I'm just going to quickly go through oat response to PKNS trial that was conducted with these 4 Agri-ARM sites. So we looked at a nitrogen response, and we looked at it when you've got the full meal deal of PKNS. And so we looked at 40 pounds of P2O5 and 15 pounds of K2O and 10 pounds of sulfur. And then we did the nitrogen response for, okay, the scenario where we take the sulfur out and we take the potassium out and then we take the phosphorus out. So if we look at the results, summarize the results for that, you can see we've got all our sites here and we have the yield potential for each one of those sites. And we have what percent increase in yield did we get from having phosphorus in the system, what percent increase did we get from having potassium in the system, sulfur in the system, and what were the soil test results for each one of those situations. So if we look at the P response first here, you can see that we have a number of sites that had a positive yield response to phosphorus. The one in green is the only one that was statistically significant. And it kind of correlated somewhat with what we found in, the soil tests. The low soil testing sites tended to be more prone to a response. And that's not surprising, while yield response to added P is correlated to soil test levels, it's not a great, perfect correlation. It's pretty messy. So it's not surprising that we get some variability here. If we look at potassium here, there were 2 sites that had a positive yield response, with one of them being Yorkton. And it was significant at Yorkton. And at first it kind of surprised me because Yorkton had very low yield potential because of drought. And that's the key word there, because of drought. And potassium often is beneficial to the crop during drought conditions. So I've seen a few times where I've got low yield potential and getting good responses to potassium, and it's mostly because of the drought conditions. At our location, many times I don't see a response to potassium under ideal conditions. So it's kind of interesting. If we look at the sulfur, again, Yorkton, for some strange reason, had a significant response to sulfur, which that really surprised me. Particularly if you look at our soil tests, they're very high. But you can't always trust a very high soil test for sulfur. Sulfur is notorious for hot spots. And you just have to hit one of those hot spots and you'll have a high soil test when much of your field may be sulfur deficient. So I think the important thing with phosphorus, just taking a step back to there, it's important to maintain your phosphorus levels high in that range of about 15 parts per million. And the reason for it is based on past studies that if you let your soil phosphorus levels get low, you'll never be able to apply enough phosphorus in season, side banded to the side, or seed placed, to make up for that loss here. And you can see that here with, we've got 3 levels of soil test N here, being 5, 8, and 15. And with the low soil testing phosphorus, doesn't matter what rate of phosphorus you put down here, you don't get a yield that's as high as the field where the phosphorus levels had been maintained at 15 parts per million. So for much of these studies, I'd like to thank Sask Oats for their involvement and the Sustainable CAP program for funding.
Thanks, Mike. That was great. Also, as a reminder, if you have any questions, please put them in the chat box. So with that, we'll get on to our next speaker, and that is Amber Wall, Assistant Manager with Wheatland Conservation Area since 2017. And her topic is demonstrating hybrid brown mustard and composite yellow mustard response to micronutrients. And we'll start whenever you're ready, Amber.
Thanks, that's right. So this is funded in part by the Government of Canada under the Sustainable Canadian Agriculture Partnership, a federal, provincial, territorial initiative, and it's also co-funded by the Saskatchewan Mustard Development Commission. For anyone not aware, Wheatland Conservation is located in Swift Current and has been a nonprofit organization that's been in operation since 1983 and was a large part of creating the Agri-ARM network. At Swift Current here, we run about 70 trials per year. We host our annual summer tour together with the Ministry of Ag. And then we also host a number of industry tours for those partners to showcase their new products or products that they sell in the area. We have 4 full-time staff, and then we hire a few local summer students as well. So this is an Adopt and Sask Mustard funded demonstration that we're focusing on today. It's looking at 2 different classes of mustard. That's the AAC Yellow 80 and the AAC Brown 18. And in this demonstration, we're fertilizing macronutrients based on a soil test recommendation when targeting a certain yield. And then we're also adding in some micronutrients on top of that to target a proper nutrient balance in order to optimize yield and see if there's any benefit to the addition of adding those micronutrients. Micronutrients are no less important in plant nutrition than NPK and S. Specifically for mustard and of interest for this project, we're looking at zinc and boron, but overall these micronutrients play a vital role in many functions across plant development. With previous demonstrations done in the SFP and ADOPT program, having provided a good proof of concept showing the response of hybrid brown mustard to increasing nitrogen rates and establishing the appropriate seeding rates, the objective here now was to demonstrate further information on the hybrid and to include that composite yellow mustard as well. So this took place at 3 locations, Swift Current, Indian Head, and Redverse. It was set up as a split plot design with having those 2 different mustard classes. Our 6 treatments are the same for each variety and range from 0% to 125% of the recommended fertilizer rate for NPK and S from the soil test results. So 0% of the recommended rate, that treatment didn't get any additional fertilizer. Next is half the recommended rate, followed by 100% of the recommended rate. And then treatment 4 and 5 have the micronutrients. So 100% of the recommended rate of NPKS plus 5 pounds per acre of boron. And treatment 5 is 100% of the recommended rate of NPKS plus 5 pounds per acre of zinc. Treatment 6 there, exceeding the limits, fertilizing for 125% of the recommended rate of NPKS. Composite soil samples of each site area were submitted for the residual nutrients and basic quality analysis. And we wanted to fertilize to target a brown mustard yield of 40 bushels per acre and yellow mustard yield of 30 bushels per acre for Indian Head and Redverse. Swift Current being drier, the Swift Current brown mustard yield of 35 bushels per acre and 25 bushels an acre for yellow mustard. So each site's actual applied fertility does slightly vary based on the yield potential of each location. So this chart here was the residual nutrients at each site. Right in the middle there's nitrogen. Each site has low to very low residual nitrogen. Probably what's considered medium phosphorus and sulfur levels at Swift Current, but even higher sulfur levels at Indian Head and Redverse, up to 90 pounds per acre. And lastly, we really want to take note of the zinc and boron here. Micronutrients are needed in quite small amounts, so this could give us some insight to how much of a response we can possibly expect. So very low boron and low zinc residual at Swift Current. Indian Head, high boron, medium zinc, not a perfect scenario for this research project. And Redverse, medium boron residual and low zinc residual. So this is a lot of operation details here on this site, but we just want to see all these sites were seeded mid to late May at 237 seeds per square meter. We do seed in plants per square foot or per square meter, so that in the case you're using a different seed lot, which is very likely, therefore a different seed size, you still know what plant stand we targeted in this project, as your pounds per acre seeding rate will not be the same as ours if your seed size is different. The zinc and boron products used were provided to us in kind by Nexus BioAg. Boron provided as Granubor, it's 15% boron, and the zinc product is zinc sulfate. It's 35.5% zinc and 18% sulfur. And both of these products are granular and were side banded along with the recommended NPKs. General conditions from site to site. As a few speakers have said here today, Swift Current did have that narrow strip of hail come through 2 days after our tour there. It resulted in some shattering. The brown mustard had higher losses compared to the yellow, and this did create some variable results. But overall, you can see the other two sites there well below the long-term average, so some hot and dry conditions. Getting into results, starting with spring plant densities in plants per square meter. The first chart here by variety is the 3 sites averaged together. Overall, brown 18 had slightly higher emergence compared to yellow 80. The exception here was at Swift Current, where variety emergence was not significantly different. And the second chart there, mustard emergence by site, so varieties together. Swift Current definitely on the lower end there, as expected, due to things like residual moisture, low organic matter, but about 70% emergence at the other locations, so pretty good overall. At Swift Current here, again, plants per square meter on the vertical axes. We've got our treatments on the horizontal axes. For both varieties, we saw a slight decrease in plant stand as fertility increased, but other than the 0% added fertility, not a lot of variation between treatments, and this trend is fairly common, becoming fairly common at Swift Current with our dry conditions. You can see here there wasn't a lot of variation between plots. This is the AAC yellow 80 along the top row here, with the brown on the bottom in order of increasing fertility. At Indian Head, the brown 18 not showing a large response to fertility, but the yellow 80 showing increased emergence in response, especially to both the boron and the zinc there compared to the 100% recommended rate. Redverse, the addition of zinc, showing an obvious increase in plant stand for the brown 18. Boron eliciting a bit of a response as well. And then yellow 80, there generally a slight increase with fertility. We didn't quite get to our target yields. This is Swift Current. We were targeting that 35 bushels an acre for brown mustard. Our yield, AAC brown 18 yields ranging from 700 to 1100 pounds per acre, so about 14 to 22 bushels an acre, but okay considering the hail. A significant yield response from the additional zinc and macronutrients. And then yellow 80 yields ranging from 700 to 1200 bushels an acre, so about 24 700 to 1200 pounds per acre, so 24 bushels an acre, getting close to our 25 bushel an acre target yield there. And a yield response to both boron and zinc, although not significantly different from one another, and the addition of the boron and zinc yielding higher than the extra macronutrients, the 125% recommended rate there, which is also, it's good to see. It could possibly be reason to limit further increases in nitrogen use, or an indication that there's a proper nutrient balance between the macro and micronutrients at that 100% soil test recommendation with the addition of the boron or zinc. Indian Head, as we recall, the medium and high zinc and boron levels in the residual soil results, not resulting in much of a yield response here from the micronutrients, other than the yellow 80 showing a small yield increase from the zinc treatment compared to 100% recommended fertilizer rate. Overall, more of a response for each variety from the increased macronutrients, which we could have expected as the soil test indicated very low nitrogen residual there. But still good yields here. Brown mustard yielding 33 bushels an acre, not quite reaching that 40 bushel an acre target, but yellow 80 out yielding the 30 bushel acre target, averaging 34 bushels an acre. Redverse, both varieties resulted in a yield response to zinc, not significantly different than the 125% recommended rate, similar to the Swift Current site. Again, knowing residual nitrogen was low, we can assume that that's a response to nitrogen. Residual boron was medium at this site, so not too surprising there wasn't a response, but the zinc treatment showing a good yield response, again, showing a potential to limit that further increase in nitrogen when you are using the proper micronutrient balance. Neither variety quite reaching its target yield here. When you average yield for all three sites, we do see a yield response with increased fertility for the most part to zinc and nitrogen, which we noted are not significantly different from one another. As for boron, it's important to note the organic matter at each site. For example, Swift Current is low, and low organic matter can contribute to low boron availability. But it's also noted in previous research that large yield responses to boron fertilization in Western Canada are rare, so we're not too surprised by these results. For boron in this study, though, the most notable response was from the yellow mustard at Swift Current, where the soil residual levels were considered very low. So other data collected in this project included lodging and days to maturity. Generally, lodging increased with fertility as expected, but wasn't too dramatic or noticeable. And the brown mustard had slightly higher lodging than the yellow mustard. And this was, for the most part, site-specific, with Redverse having the highest lodging incidence and Swift Current showing very little. Maturity was largely affected by drought and high temperatures, so ended up being pretty early at each site. As well as the hail at Swift Current made this quite a difficult rating when pods you expect to fill don't, but there ended up being no significant difference between varieties when all sites are combined. 1 to 2 days longer to mature at Indian Head and Redverse with increasing fertility. So to close, even with the low yield potential for the year, we're pretty happy with these results. There was a response to zinc in most cases, with the exception of the brown mustard at Indian Head. And contributing factors there were that medium zinc residual level, as well as the limited moisture. Not a strong response to boron, but we did see yellow 80 have a small yield increase at Swift Current, the only site where levels were low. And as expected, a response to added sulfur and nitrogen where residual levels were low as well. As we know, getting a high yielding crop takes a number of things to go right, but this study has demonstrated that soil testing can be very important. Just like NPK and S, mustard will respond to proper micronutrient management as well. So first and foremost, get your macronutrients right, followed by your proper seeding rate. But we can see here that micronutrients should be considered in the overall picture. One, to avoid over applying that nitrogen, and that's where that composite soil test is very important. and secondly, to optimize your yield and economic return. I want to give a big thank you to Corey Jacob, Shannon Chant, Rick Mitzel. They're the ones that put this project together and applied to the adopt program, and Sask Mustard for their co-funding contribution as well. Mustard 21 for providing us with the seed for the project, and then Nexus BioAg for sending their zinc and boron products over. That was a huge help, as well as the other 2 participating sites and their staff. And of course, Wheatland. We've got quite a crew here that none of this would get done without. So this was just a 1-year demonstration. The final report will be up on the website once approved.
Thanks so much, Amber. The next speaker that we have is our very own crops extension specialist, Kim Stonehouse, who works out of the Tisdale Regional Office, and he will be talking about the risks of top dressing nitrogen in the wintertime. So we'll start whenever you're ready, Kim.
Okay, thank you so much, Eileen. As mentioned, my topic is top dressing nitrogen on fertilizer and on frozen and or snow-covered ground. This was an SFP project that was designed and took 2 years to complete. I'm going to just try and get this out of the way here. Okay, there we go. So a little bit of background about this project. There are high nitrogen rates that are generally being applied at seeding, and they can significantly slow down operations due to the extra time required to haul and fill the seeder tanks. And as well, there is a added labor requirement and increased capital investment for trucks to haul the product to the field and seeders capable of banding it. So in response to these logistical issues, producers have looked at ways to apply nitrogen fertilizer at different times of the year. One of these practices involves the broadcasting of granular nitrogen fertilizer onto cold or frozen soils, often covered with snow. There is some information out of the US that suggests that this may work in specific situations under specific conditions, but there's really no Saskatchewan specific data, and we know that our climate is different. So the question came up as to how we advise producers about this practice. See, the objectives were simple. The objectives were focused on basically the potential for losses in production and the economic risks associated with broadcasting applications of nitrogen on frozen and or snow-covered soils. This is, as I mentioned, is a strategic field program study that was conducted over 2 years and was funded basically through the Sustainable CAP program. The bottom line here really is that we know that this is happening, but it's very unlikely that the practice is environmentally or economically sound. The study occurred at 7 Agri-ARM sites. Brianne McGinnis of the Northeast Agricultural Research Foundation was contracted to coordinate the project. The sites included the CLC at Prince Albert, ECRF at Yorkton, ICDC at Outlook, which was our only irrigated site, the IHARF at Indian Head, NARF at Melfort, SURF at Redvers, and the Wheatland Conservation Area at Surf Current. And this was done basically to look at and assess the practice under different soil zones and climatic conditions. Methodology was fairly simple. The study was set up as a randomized complete block design with 4 replications. The nitrogen application rates were based on typical target yields for the area and the soil test results, of course. Phosphorus was maintained at 34 kilograms per hectare of P2O5, and hard red spring wheat was chosen as the indicator crop for this particular study. The treatments consisted of 3 nitrogen sources. There was urea, then a urease and a nitrification inhibitor, and a urease inhibitor on its own. There were 3 application timings, mid-November, early February, and early April. And these, of course, were compared to the side banding treatment at seeding. Data collection consisted of the fall soil test and recommendations, the fertility rate, application dates, soil temperature at application, the snow depth at application, meteorological data, both in the wintertime when the fertilizer was being applied, as well as during the growing season when the crops were growing, the seeding rate and date, the crop variety, the seed and nitrogen costs, and then our comparative factors would be grain yield and grain protein. Conditions at the time of applications. During the first application, 7 out of the 14 site years were frozen, so about half the sites. And during the second application, all sites were frozen. And during the third application, 1 out of the 14 site years was frozen, and it also was the only one with snow cover. So snow cover ranged from 2 to 30 centimeters at the first application date. Snow cover ranged from 9 to 49 centimeters at the second application rate, and most had 0 snow on them, except for 1, as I just mentioned. Conditions, of course, what happens when you decide to conduct a study where you want nitrogen to be your limiting factor? Of course, then we have a drought. In 2021, 4 of the sites were well below the long-term average precipitation, and 1 site was significantly below. In 2022, 2 sites were below the average, and I would say well below the average, and 1 site then was well above the average where flooding occurred. Nitrogen rates, of course, were chosen to be a high target yield, and 4 of these sites actually had high soil residuals. So I think, as you can imagine, drought and high application or nitrogen residuals can mask fertilizer losses when comparing yields, but it's less so for proteins, as we'll see later on when I start to discuss the results here. So I have a table, and I put it in as a table because the graph kind of gets a little bit messed up, and this is just the first year results. But I know there's a lot in here, but I've kind of highlighted the maximum yield so that you can get a sense of the pattern that was developing. So grain yields were measured for each plot and adjusted to 14.5% moisture content for both years. Results were statistically compared through analysis of variance with multiple comparisons of means. The coefficient of variance, the least significant difference for the 95% confidence interval, and indication of significance is presented here. And the results indicated basically that 3 out of the 7 sites in the first year, 2021, found significant differences, but they were not between all treatments. And likely, again, they were masked by some of the drought that was going on. The greatest frequency, though, of top yields was the side banding treatment at seeding. And the 4 out of the 7 years had the highest yield with the side banding treatment. The drought masked fertilizer responses at the Conservation Learning Center, CLC, and East Central Research Foundation, and NARF. Moisture was not the limiting factor at ICDC since it was under irrigation, and this led to the highest yield potential for all site locations. In this case, it's double, just about what every other site produced. Oops, sorry about that. In 2022, we saw similar results. Again, 3 of the sites showed significant differences, but again, not all between all treatments. And again, the greatest frequency of top yields was with the side banding treatment, 4 of 7 of the sites. So a total of 8 of the 14 site years. One site, at Agri-ARM, indicated significantly greater yields was achieved for this side banding treatment over all other treatments. At the NARF site, the early April application was missed, and therefore the values in red were not included in the analysis. Again, the drought and high application of residual rates of N masked the nitrogen losses at the CLC, East Central Research Foundation, and the Northeast Agricultural Research Foundation. And you'll note on this slide that the flooding at the Redverse site at SURF increased the coefficient of variance to beyond 20% and reduced the number of the replications. So this data was also omitted from the final analysis. I just put this up to get an idea of what the average was over all the 14 site years. So I averaged all the yields, and on average, the greatest yield was achieved when the nitrogen was applied at side banding at seeding, 8 of the 14 sites. Applications made in early February had the lowest yields, regardless of whether an enhanced efficiency fertilizer product was used or not. The enhanced efficiency fertilizer products showed some benefits over the untreated urea for mid-November and early April, but did not equal the benefit of side banding. A unique result here is that the bare urea treatment showed an average yield for the mid-November, early February, and early April of almost the exact same amount. So I don't know whether this is a unique factor or not, but it was kind of something unique I thought I would point out. When we look at protein differences, protein was a little bit of a different story. In 2021, 6 of the 7 sites showed significant differences in protein levels. Of course, again, not between all the treatments, but there were quite a few more than what we saw in yield. 5 of these 6 sites showed significant differences that indicated the maximum protein was achieved when the nitrogen was side banded. In these cases, the side band was not always significantly better, as I mentioned, than the other treatments, but it was consistently almost always significantly better than the early February treatments, and often significantly better than the mid-November treatments. In 2022, again, 6 of the 7 sites showed significant differences in protein levels. 4 of these sites indicated that there were the significant differences had the maximum protein was achieved during the nitrogen side banding at seeding. And again, the side band was not always significant, but better than every other treatment. Sorry, it was not always significantly better than every other treatment, but it was under the irrigated site. So that's kind of an interesting note. An explanation of why there were more site years that achieved the highest level of protein for side banding than the yield is that protein is built at a much later time in the plant's life cycle and is dependent on the amount of nitrogen available at that time. This indicates that even though nitrogen losses were masked by the lack of moisture when considering the yield, they are still occurring and are indicated in the protein levels. Again, I averaged the results over the entire 14 years, and overall, the average protein was the greatest when the nitrogen was applied at seeding at 9 of the 14 site years. Applications made in early February had the lowest protein, regardless of whether an enhanced efficiency fertilizer product was used or not. And again, the enhanced efficiency products may have shown a small benefit over the untreated for the mid-November and early February, but these did not equal the amount of increased protein that you could achieve through side banding. Finally, I want to talk a little bit about the economics. So the economics for this particular project were determined for each treatment using the difference in the mean yield of each broadcast application relative to the mean yield achieved for side banding, and then multiplied by the current sale value of CWRS. That value turned out to be almost $400 per ton, 397. And in addition, the broadcast applications incorporating an enhanced efficiency fertilizer product had an added cost for this product, and so that was also subtracted. This premium is subtracted from the amount to give a more representative example of what the losses could potentially be. And these results indicated that on average there was an economic loss for all broadcast treatments. Enhanced efficiency fertilizer products reduced the loss for the mid-November treatments slightly, but increased the cost for the other timings due to the premium that was paid. So the take-home message from this is that dry conditions and the high variability in high availability of nitrogen masked nitrogen losses. Adequate growing season moisture showed significant yield benefit to side banding. The nitrification and urease inhibitors showed some potential benefits, but they were not consistent for the top dressing and did not fully restore the yield or protein potential that could be achieved through side banding. Significant grain protein benefits were seen for side banding when compared to top dressing, and all of the broadcast treatments had an economic loss when compared to side banding. And that's it for me. I just thought I'd put my contact information up here. If anybody wants to contact me and discuss the results any further, they're also posted online or can be found through the Ag Research branch and obtained that way. Thank you very much.
Thanks for all that great information, Kim. At this time, we will have all of our presenters turn their webcams on, and we will begin our question panel to chat more about fertilizer management. And with that, I'll start off with the first question that we have, and this one's for Jessica, and that is, if producers were trying this product, how would you suggest they go about it? It says apply to the whole field or do check strips?
That's a great question. I would strongly recommend going with a check strip. At this point in time, we know that the efficacy is a little hit and miss. So I think your best bet would be to use a check strip on it, a little area, so you can see compared to your regular fertilizer applications how it compares. And then once you get a little bit more comfortable with the product, and if we can really hone in on our application timing to get a yield boost with this product, then I would recommend maybe going all in. But at this point, I would just be more on the conservative side.
Great, thank you. And another one for you actually, Jessica. And that is, did you attempt to use drop nozzles and spray the bacterium to the underside of the leaves where the stomata exists on the leaves?
We did not. We just used a regular flat fan like you would use in any other typical spraying operation. One of the things that we did learn with this product is it does enter through the stomata, but it doesn't necessarily have to go through the stomata, just the way that both bacterium are able to penetrate the leaf cuticles. They don't have to go through the stomata, so we didn't necessarily go through that route. Just for ease of application, we wanted to mimic how a producer would do it.
Great, thank you, Jessica. We have a question for Brianne, and that's, why does high nitrogen rates reduce test weights?
I don't know if I can necessarily answer that. If I know the reasoning for that. I know sometimes for us it seems to be a factor of yield, but I don't know if someone knows the actual science behind that more than I do. I just know it's something to consider when pushing your nitrogen rates.
Thanks, Brianne. If anyone has anything to add to that, any of the speakers, just feel free to jump in.
I have asked Bill May that question in the past, and he says we don't really know. My thought was that, you know, as you increase nitrogen rates, you increase the amount of tillers that also start contributing to the yield, and I thought it might be related to that. And he says, no, we actually don't really know. So I don't know. We're looking for hypotheses, I think, for why it happens, but it is a well-documented phenomena that occurs all the time, basically.
Thank you, Mike. And the next question we have is actually for you, Mike, and that is Summit the better option to grow because of the test weights? And what factors should producers think about when choosing varieties?
No, I wouldn't say Summit is a better one, because the thing is, like, Camden is always at the bottom of the pile for test weights, and yet it makes up like 50%, you know, of the oats that are accepted by grain millers. So there's a bit of a disconnect from what we see in our plots and what's influencing producers in the field. And it's been suggested to me that in our plot work, we may hang on to more of the fine seeds than what producers are doing in the field, and they may be blowing more light seed out the back of the combine. And so when I've talked to grain millers about it, they rarely have an instance where they've, you know, said, no, this isn't going to make the grade. They usually can work with somebody on that. The thing about Summit is that Summit doesn't have as good a lodging resistance as a Camden. And so despite having huge, much larger test weights, I wouldn't pick Summit because of that, because it's not proving to be a problem for the producer for Camden. As far as what you should pick, I guess, depends on what issues you've had. If you've got lodging issues, and boy, you got to keep with something that's got good lodging resistance. But obviously, you sort of pick from the end user what their what's on their preferred list. You don't want to be growing something that, if you're going for milling and it's not on their list. So choose something that. Probably lodging resistance is a key consideration for a lot of people, particularly if you want to start pushing the envelope for nitrogen rates out there.
Thanks for the detail, Mike. We're now going to turn it over to Amber. And the question is, how did you determine the yield potential for each of the sites that you were basing nutrient needs off of?
Yeah, so those soil tests were sent into AgBuys, and then most companies that you send a soil test will you can put a target yield in there, and then they'll send back what you should fertilize. So I guess the mustard growers, Corey Jacob, they got together and they picked that yield target, and then you input it in your soil test and they'll tell you what to fertilize.
Great, thank you. Another one for Jessica, and that is, was there no response at Scott or Prince Albert because of the low pH?
In terms of Prince Albert, I think the underlying factor for their lack of response was because they had really high residual nitrogen levels. So I think that was more of the factor than their pH. And at Scott, truthfully, I'm really shocked that we didn't have a nitrogen response. In many previous studies where we worked with our nitrogen trials, we do typically have a nitrogen response, and our soil pH hasn't changed from previous years where we have a response. So truthfully, I'm really shocked we didn't get a nitrogen response at Scott, and I really don't know why it occurred this year.
Thank you. We have a question for Kim, and that is, dry conditions may have impacted results, would you consider doing this project again in wetter years to see how that impacts results?
I think that's a great question. Yes, this is something that it ideally it would have been nice to have a situation where moisture was not the limiting factor. Unfortunately, you can't do everything under irrigation. It would be nice to see years where we had more moisture, and then we could just test the nitrogen and to see whether or not we're going to get results from it or not, and whether broadcasting would be that much worse or what have you. But I mean, we can't pick the year that we're in. So yeah, I would love to see it done more.
Absolutely. Thanks, Kim. And a question for Brianne. Based on the side banding versus mid-row banding results, are you expecting these results to be different within different soil zones and environmental conditions, like down south?
Yeah, I suppose it could be. I mean, moisture would definitely be a limitation just in overall for the nitrogen response. Yeah, it very well could be. If I'm thinking back to individual site years too, I think it was last year at Melfort where we had a lot more moisture, where we had more of the significant response to side banding versus mid-row banding. So it's potential that just moisture conditions and your soil conditions and your environmental conditions are going to affect the likelihood of a response. So
Thanks, Brianne. And Jessica, is the nitrogen fixing research now complete, or will it be done again? And they go on to ask, are there any plans to do economic analysis on all the research involving nitrogen or potential nitrogen enhancement products? And what is the return on investment or best bang for your buck? So a bit of a three-parter there. I can
No
repeat
problem.
anything.
Sure. So the first part is, are we going to be repeating this trial? We have put in for funding for to repeat this trial on canola and wheat. We're just waiting to see where that's going to lie. We still have to talk to the commodity groups and just figure out if they're able to fund these kind of projects again. I know that there's quite a bit of interest among the Agri-ARM sites to do this work once again. And we do want to speak to the product suppliers just to see if we can really fine tune the application process. So we certainly are hoping to, but we can't confirm it yet. What was the second question? The economic analysis?
Yes, absolutely. Are there any plans to do economic analysis on all the research involving N or potential N enhancement products?
Absolutely. I think that would definitely be the next step. Whether or not we see a result if we repeat this again, I think there certainly will be an economic component, because I think when it really gets down to it, we need to see if this is a profitable option for producers. So I certainly think there's going to be an economic component going forward. And then I think, what was the last question?
Absolutely. What is the return on investment or best bang for your buck?
At this moment, I would just continue to recommend using your urea for all of your regular fertilizer operations. At this point, putting on this product and having to put it on, at a separate time, and really having to hone in on your application timing, I just don't think it's really economically feasible at this moment for producers, unfortunately. But I do think going forward, once we're able to fine tune it, you could get quite a bang for your buck if you're able to actually reduce the amount of nitrogen you have to put down. But currently, I would just be going with your old-fashioned urea or any of your enhanced efficiency products, and just keep doing what you're doing until we can really fine tune this process.
Great. Thank you, Jessica, for all that detail. And that actually concludes our question panel for today. So at this time, we would like to thank everybody for taking the time to join us for the event, and all of the presenters for providing such great information. And don't forget, for all those who are CCAs, the QR code will be appearing on your screen